So, one of the ways to effectively transport oxygen means that we need to vary our affinity. That means a protein has to have essentially different states, different curves. It has to have a state where it's very high affinity in certain times so that it can bind oxygen even when there's very little amount of pressure. So very, very, very rapid binding of oxygen, for example, when it's in the lungs. But when it moves to the tissue, we need suddenly this line to drop, right? We need the ability of it to hold on to oxygen at that point to be low because we want the oxygen to now go into the tissue. But how can you change the affinity of a protein? How can you make it suddenly have high affinity and then low affinity? Two different states, essentially. Well, the way to do that is to have a protein that has multiple binding sites. And each of the binding sites has an ability to change the protein's interaction with the next binding site. This is a phenomenon that we call cooperativity. So, again, for example, you have 4 binding sites. When all the binding sites are empty, let's say that this particular enzyme suddenly has very high affinity for its ligand. So, a ligand comes around and it binds really fast, really high affinity. Once that first ligand is bound, this protein will shift and it will actually change its shape a little bit. And these sites that are now available will actually be a little bit less receptive to binding. But let's say another ligand comes around and eventually it binds to the second site. Now, what will happen yet again is that the binding now of this second site will affect the 2 others around, causing them to be have a little bit less affinity maybe for the ligand. This is called negative cooperativity. This is where the first binding reduces the affinity of the remaining sites. Now, of course, you could actually have the opposite happen. Once the ligand binds one site, it might make it so that these sites are even more likely to bind ligand. So they'll even more quickly bind, like that until they are completely full. This is called positive cooperativity, and that means that the first binding effect increases the affinity for the remaining sites. We're going to talk about what this would look like on the Hill equation. If we plotted this, how would a protein that has cooperativity look like? Remember, you'd kind of have different states. You'd have a high affinity state and a low affinity state of the protein. And it would change as you bind. So we're going to talk about that actually in the next lecture. We'll go more into cooperativity and again, looking at other types of ligand interactions in relation to proteins and more case studies and examples of protein interactions with their ligands. I'm going to elaborate more on cooperativity, which is, again, when you have the changing of a structure of the actual protein after binding of a ligand. So here's an example of cooperativity in a dimer. This is a protein that has quaternary structure. So there are 2 subunits that make up the whole protein complex. And you can see this does not have anything bound to it in the first panel. And there are areas of its binding site that are open for ligands and other areas of the protein, which are highlighted in red here, which are described as being unstable.
Once the ligand binds to one of these binding sites, you can see that there's actual change in the conformation of the protein. And the change in the conformation of the protein, usually in one subunit of the protein, can thus actually change the affinity for more ligand in other binding sites on the same complex. And again, this is called cooperativity. And if you remember, cooperativity can be positive or negative, meaning that the change that occurs based on binding can affect other areas of the protein in a way that makes it more likely for a ligand to bind or less likely for a ligand to bind. In this example here, it's showing positive cooperativity in that the initial binding, which stabilizes this binding site, makes the other areas have higher affinity for the ligand. So this is a positive cooperativity example. Cooperativity is an example of what we call allosteric regulation. And this is something as we get way further into Biochem 1, when we're going to talk more about pathways and protein pathway interactions, we'll be talking a lot about allosteric regulation. Allosteric regulation, allosteric protein is, again, binding of a ligand to one site that affects the binding properties of a different site of the protein. So this, again, is the definition of that cooperativity. And again, it can be positive or negative. Now, it can also be homotropic, which is the normal ligand of the protein is the allosteric regulator. So this just means that the ligand that normally would be binding to the protein is also what regulates the protein. It can also be heterotropic, meaning that a different ligand, so some other molecule, affects the binding of the protein for whatever the normal ligand is. And I'll give you two examples of that very quickly. Say you have a protein, and in fact, one of the ones we'll be using as our example is hemoglobin. Hemoglobin has these 4 subunits on it. Now, the ligand of interest in hemoglobin is oxygen. So when oxygen binds, it makes it more likely for other oxygens to bind. This is what we call homotropic. Now, there are some proteins, and we're going to be seeing these a lot when we talk more about allosteric regulation. They still have cooperativity, but they're heterotropic. So, for example, some proteins like kinases have a binding site for ATP. This is another molecule. And when they're bound to ATP, it makes them more likely to bound their main ligand of interest. This could be something like glucose. So this is heterotropic. That means some other molecule essentially will bind and either activate or deactivate the protein for its normal job with its normal ligand. So in this case, the normal ligand would be glucose. This protein, let's say, helps break down glucose, but it's ATP binding that controls whether it is activated or deactivated. So again, these are all types of cooperativity, either positive or negative, which we also can call allosteric regulation. This second type, the heterotropic type, again, we'll be seeing this a lot, which is a way that biological units, and in our body, it's a way that we regulate pathways. Something like ATP or some other ligand can activate or deactivate our enzymes, our proteins, to do their job. Now let's go back to our example of hemoglobin. If you remember, we left off talking about hemoglobin and how it has multiple subunits. It has this heme group in it that binds oxygen, and it is having this positive cooperativity. It's our example of positive cooperativity, and we're going to look at how that affects um, its binding curves, etc. So this is just an example of the structure of hemoglobin and myoglobin to show you and kind of get an idea that myoglobin is very structurally similar to one of the subunits of hemoglobin. And hemoglobin is made up of 4 subunits. You can see it has 2 of these, what we call alpha subunits. These are separate peptides, remember, separate peptide chains that have folded into a quaternary structure. There's alpha 1, which has essentially the same structure as alpha 2. And then beta 1, which has the same structure as beta 2. And the alpha and beta are different from each other. You can see here that the beta subunit of hemoglobin is almost identical structurally to myoglobin. Now, myoglobin does not have these 4 subunits, it just exists as one folded protein. So there's no quaternary structure for myoglobin. And remember, myoglobin is very good at binding oxygen and holding on to it, but it's just used for oxygen storage. And remember, that's because if we look at its hill plot, it stores oxygen very, very, very tightly, but it's not able to then release oxygen in different environments when you compare the lung or tissue. So we need something that's able to bind it in certain conditions and release it in other conditions, which is why, again, cooperativity is very good in this case because the 4 subunits of the hemoglobin act together to make the whole molecule either more or less affinity to oxygen. Again, this is just some subunit interaction to give you an idea of what hemoglobin looks like as far as these 4 subunits. So each of these chains here that are kind of blocked off, this represents one string of amino acids for that subunit. And you can see it has its amino end and it has its carboxyl end, right? And here, all of this diagramming is just showing you different types of tertiary interactions that are occurring either within a chain or between chains that are holding the chain together. So, for example, you can see that this beta 2 chain and this alpha 1 chain have some type of ionic interaction between the carboxyl end that's negatively charged and this lysine group that is positively charged. You can also see there's ionic interactions between the alpha 1 chain and the alpha 2 chain. Here's an arginine, an aspartate, and also another aspartate and arginine oppositely paired that are interacting ionically through the alpha 1 and alpha 2 chain. So this is showing how these non-covalent interactions, these tertiary interactions, can help hold together quaternary structure of different subunits to make a whole complex. Now, this cooperativity in hemoglobin has been very well described, and we have given the names of the 2 states of hemoglobin when it is either in its low affinity or high affinity state. the T and R state. T stands for tense state. And in this state, it actually has more interactions, more of these subunit interactions that are shown here. And because of the interactions that are going on between the subunits, it actually has lower affinity for oxygen. And that's just because the areas where oxygen would bind on those heme molecules are less exposed. So tense state, it's very tense and has lower affinity for oxygen. The R state, or the relaxed state, there are fewer interactions, it's more flexible, it's more open. Those heme molecules are open for binding to oxygen, and so it has a higher affinity for oxygen. Remember, when we're saying higher and lower affinity, that means that the actual KD, that actual number, that constant number of how likely it is to bind, at what concentration it will bind, is changing. So the change in the conformation from T to R state actually does change that KD number. It changes how likely it is to bind oxygen. Now remember that in this case, the molecule has cooperativity. And for hemoglobin, oxygen binding triggers the molecule to go from a T state to an R state. So once it does bind oxygen in its T state, it's more likely to bind oxygen, right? So it will go from T to R state. And that is again due to conformational changes that occur. Once one oxygen molecule gets in there, the whole molecule has to shift a bit and it becomes more and more like the R state, in which it has a higher affinity for oxygen. And here's just a look at those two states. You can see some changes in the three-dimensional structure of the T state or tense state versus the R state. You can see the R state actually looks a little bit more expanded. Things are kind of sticking out more. It's taking up more room. And again, that goes with this idea of it being more relaxed, it's more open. Those heme molecules are more available to binding oxygen. Here's a closer look at a conformational change that is occurring that actually is shifting and triggering oxygen binding. So here's a close up of the heme molecule. Remember, heme is held in this state by a histidine. You can see it right here. And here it is bound to an oxygen molecule. Here's the heme bound to an oxygen molecule versus not bound to an oxygen molecule. And you can see actual shifts in the relation of where the heme is to where the other amino acids are. So this slight conformational change of the movement of these surrounding amino acids actually shifts the other subunits within the molecule. Now, here's the big thing. How does cooperativity affect what we were talking about in plotting the hill plots? Well, to add in the information of subunits, to add in mathematically the idea that we have subunits present that are um, acting either together positively or negatively, our equation needs to change slightly. So we've already seen these two equations. This is our Ka equation, the equilibrium equation, which shows the products over the reactants. And this is just that very general equation for the ratio that we're using, which just shows ratio of the ligand to ratio of the, to the ligand and the KD. And this equation right here is what we use for our Hill equation, right? It has our ratio number, our concentration of ligand, and our KD. And you'll notice now that this N has appeared, and the N is just the number, this number of cooperatively participating subunits. The N is going to change the shape of this hill plot. And you can calculate the N if you have your hill plot and you plot the shape of it. You can calculate the N. But in this case, I'm just showing you how a different N value will affect your plot. Now, in our example previously with myoglobin, remember myoglobin is a single subunit, and so it does not have any cooperativity. When something does not have cooperativity, its N number is 1. And you can see if you plug 1 in here into the N, that's just now L to the 1, which doesn't change anything. And so we get our original equation that I showed you, which was just regular concentration of L over L plus KD. So when N equals 1, no cooperativity, and you can actually see what does that look like on the plot here. It looks like this parabolic shape that we already saw in the previous example for myoglobin. Now, what happens if you change the cooperativity to either being more positive or more negative? Well, when you have a more positive cooperativity, so for example, our N equals 4 here. So when N is greater than 1, you have positive cooperativity. And you see you actually have this more sigmoidal shape. Now, this is perfect for our example of hemoglobin. Remember, in hemoglobin, we wanted to have something that has, at certain concentrations, a slightly different relation between ligand and the fraction of bound to at higher concentrations. And we achieve that by getting this sigmoidal shape. So the sigmoidal shape is showing that at very low free ligand concentration, you don't have that much binding, but it quickly changes at the halfway point. And at high, very, very high, you have very high binding. So it's less of an on-off switch right here. When you have a very, very steep change, it's more on or off. And you can see oppositely when you have negative cooperativity, the fraction of binding, for example, here is going to have a very low Hill coefficient, which again changes its shape to be right like this. So here you have very, very strong binding, even at low ligand concentration. But then you can see that it just peters off at the halfway point. You never have complete binding. And that's because of this negative cooperativity, meaning that once binding occurs, you're less likely to bind. Instead of with positive cooperativity, which is once binding occurs, you're even more likely to bind. So that's how you would read and how you would note changes in a Hill plot based on it having this positive or negative cooperativity. Generally, what you want to be able to see or understand or interpret is how the shape is related to it having positive or negative cooperativity, or essentially no cooperativity. Now, we talked about how cooperativity is going to affect this Hill coefficient and the Hill plot here, which is comparing the amount of ligand to the fraction of bound protein. For hemoglobin specifically, another thing that does affect its ability to bind and its binding association, so its association constant or dissociation constant, is the pH. And this again might make intuitive sense. And we can think about how oxygen and carbon dioxide are related in pH in the blood and how that will then affect hemoglobin in the blood. So remember, we did talk about the blood buffering system, right? We talked about the bicarbonate blood buffering system, in which CO2, when we have excess CO2, we're going to generate more protons, which makes our pH of our blood lower, essentially making it more acidic. And because the hemoglobin actually is affected by the acidity of the blood, the amount of carbon dioxide around will also affect the ability of your hemoglobin to bind oxygen. So you can see that protons actually will bind to hemoglobin and they stabilize the tense state, the T state. Remember, in the tense state, you actually have lower affinity for oxygen. So this leads to the release of oxygen in the tissue. This is a good thing. I know at first it sounds like, oh no, protons binding to your hemoglobin puts it in the T state, and that means it has less affinity for oxygen. The reason that it reacts this way again is because if you are suffering from too much carbon dioxide, you start making your blood more acidic. Those acid protons will then bind your hemoglobin and make all of the hemoglobin that has oxygen in it release the oxygen. That's good for a burst of oxygen release into your tissue to help in that moment when you have too much CO2 in releasing the oxygen. Obviously, if this continues, if you continue to have too much CO2, you continue to have too much protons, you've already now released all of your oxygen, all of your hemoglobin is going to stay in the tense state. Now, in a prolonged situation, that won't be good because now hemoglobin, it's harder for it to bind oxygen when it gets to the lungs and now bring it to the tissue. So you can see the actual pH effect of oxygen binding on hemoglobin. And it is related to the amount of CO2 that is around. But also, there are slight pH changes when you look at differences in the lung and the tissue. So, for example, we know that in the lung and the tissue, it's not just a steady state of 7.4. You can see here that at a pH of 7.2, we'll go over, our halfway binding is at a pressure of around 4. Whereas at a pH of 7.6, right here, and our pressure is at this lower point. If you remember from our previous discussion, the pressure of oxygen in the lungs is actually higher, whereas the pressure of oxygen in the tissue, the concentration of oxygen in the tissue, is lower. And so by shifting the pH, You also have the ability to shift the concentration of oxygen that is bound up in hemoglobin. And so you would see that at this lower pH, you have oxygen that is going to be bound to your hemoglobin only at higher concentrations, at higher pressures. Otherwise, it won't be bound. Whereas if you have a higher pH system, you're going to have hemoglobin that is bound up only at pressures that are now lower. So it'll continue to stay bound even at lower pressures. So by shifting the pH in your lung versus tissue, you can shift how much of that oxygen is going to remain bound. So this is another way that the system and the body can affect the binding ability of that particular protein, not just through cooperativity, but also just through pH changes. So think about which setting would be better for lung versus tissue. Would you want your lung to have the higher pH or your tissue to have the higher pH? And would you want lung or tissue to have the lower pH? Another way that hemoglobin and carbon dioxide are related is through the formation of a carbamate. So about 15 to 20% of CO2 actually gets converted and released in the form of this carbamate. You can see CO2 reacts with an amino acid, that is the terminal amino acid on some peptide or protein, and forms this carbamino, essentially residue. So you have some peptide, and now your CO2 has kind of bound and reacted to the end of a peptide. And in this way, we can release CO2. This, when the CO2 carbamino occurs on a terminal residue of hemoglobin, it can actually affect salt bridges within the hemoglobin itself. And again, it stabilizes the T state. So this is another way, not just through the yielding of protons, which we talked about in this slide, in which protons affect stabilization of the T state, but also, thirdly, the actual binding of CO2 to hemoglobin can also stabilize the T state. Again, this is going to help release oxygen into the blood, but it makes the whole hemoglobin molecule have less affinity for oxygen. Now, lastly, I'm going to talk a little bit more about the molecule hemoglobin and other ways that interactions of hemoglobin can affect a system. And in this example, we'll be talking about sickle cell anemia. So this is not an example of cooperativity, but a different example of protein binding affecting a system. And again, it just happens to be using hemoglobin as the example. Now, sickle cell anemia is due to just a single mutation that affects the hemoglobin protein. It takes a glutamate number 6, and changes it to a valine. And remember, glutamate is a negatively charged amino acid, and valine is a nonpolar amino acid. This is in the beta chain of hemoglobin that it happens. The valine side chain, when it is added in, it actually can bind to a different molecule, whole complex molecule of hemoglobin. That means Just changing this one amino acid allows a whole hemoglobin complex to now interact with other hemoglobin complexes. And ultimately, this sickles the red blood cells. They get this kind of jagged shape. Of course, this is in individuals who have sickle cell anemia not good, because if you have too much sickling of the blood cells, they essentially will rupture and die. And so you have an intense anemia that occurs because of the sickling of the cells. That's the name, sickle cell anemia. So you're very low on your blood cells, red blood cells. The reason for this mutation, if you're unaware, the reason that this mutation, even though it seems to be very damaging, has persisted is because having a heterozygous situation in which you express in only one set of genes this mutation, but not in your other set of genes, you are more likely to be resistant to malaria. So, individuals who are in areas of high exposure to malaria are more likely to develop this mutation because it was positive for malaria. Though, if you have a homozygous, if you have both genes expressing this, you generally die at a very young age due to intense anemia and loss of blood cells. How does sickle cell anemia actually occur? So, as I just said, the mutation actually allows for two separate whole hemoglobin complexes to then interact together. And you can see they start to bind through their beta chains. That's where the mutation occurs, in the beta subunit. And now the beta subunits of two different hemoglobins will interact, and they start to form what you're probably familiar with, these fibers. We talked about fibrous amino acids, and fibrous proteins. Hemoglobin goes from a globular protein and starts to form these interactions that makes it then a fibrous protein and starts forming strands. So instead of acting as this single subunit, it now starts to bundle, it forms strands, and this fiber then becomes obviously very strong and crystalline and starts to just very plainly poke through the cell. So you've now made these very strong fibers within the cell that then make the cell become very jagged as to the long, strong fibrous material that's in them, and will potentially and eventually rupture the cell.